Thanks to the organizers for bringing me out today. And so this is the first of a couple of talks I'll give today talking about the SAXS experiment. And this is probably the most important part. Preparing your sample before and understanding it well before you put it into the beam. How many of you are structural biologists? X-ray crystallography, NMR, cryo-EM, all right? And so I'm a crystallographer by training as a graduate student, but over the last 15 years I've done a lot of solution biophysics hand in hand with that. And there's a big difference in culture between people who practice different methods, right? If you're an X-ray crystallographer, if you're a cryo-EM person, it's like to shoot first and ask questions later, right? You don't really need to understand your sample a whole lot to do an experiment and get a result in spite of yourself, right? Small angle scattering is very different. If you don't have a lot of a priori knowledge accumulated before you put that sample into the beam, right, you may very well mislead yourself into a solution that is not correct. Big danger. So your approach to how you apply this technique is very important to realizing success. Some suggested reading here. There's a wonderful amount of literature now versus 15 years ago, especially review articles talking about some of these very things I'll touch upon now, and a lot of practical points in terms of designing your experiment. I highly recommend these articles and others that are now available in review. All right, so just to review, this is the experiment we're interested in here. We're taking a sample that we understand well, or reasonably well, and via elastic scattering, collecting the resulting scatter from a monochromatic beam. All right, or some beam source is either a rotating anode X-rays, right, or copper alpha, or synchrotron X-rays, or you can actually use even experimental neutrons, which we're not going to talk about a lot today, but neutron scattering is something that I enjoy talking about as well. The scattering you see here, which is, basically the particles are an average in three dimensions, right? This is seemingly uninteresting, but there's a lot of information buried in there as you move away from the beam stop. That is, as you go farther out in scattering angle. That decay in the intensity, right, contains a lot of information about the size, shape, volume, molecular weight, compactness, flexibility of your macromolecule, right, what it's doing in solution, right? And so what scientists do, they take this information and they rotationally average that intensity, the function of the scattering angle, to get these one-dimensional profiles. Right? They're not spectra, they're profiles, we call them, right? And you can see how the intensity is decaying on the log scale, right, as a function of Q, or S or H, like noted, right, as a function of scattering angle. That's where the information is. Now, a more intuitive way of looking at this data would be to do the inverse Fourier transform, going from reciprocal space to real space, and getting what is called a shape distribution function, okay, which is basically a very nice description of all the possible intertonic vectors within that scattering volume, all right? And so it's a very nice readout of size and shape, peak being somewhere near RG, and the least common vector in that scattering volume being the maximum dimension, right? And so for many years, this was the experiment, and it was very useful. There's a wonderful amount of literature in the 1970s where this is applied to things like the ribosome, right, and chromatin, all right, so fundamental applications. But really, in the past 2 decades, where the technique has really taken off in its application in literature, all right, is because of the availability of personal computing, or laptops, right, and computers in the lab setting, and these algorithms that allow us to take this information and, in an ab initio fashion, reconstruct shape, right, the scattering volumes that are consistent with the experimental data. This is very low-resolution data, right? So what we do is we take this information and we reconcile it with other a priori information. The TONC models from the protein data bank, now there's 140-something thousand of them, 150,000, right? And other information we have about our system. Focus on these other solution measures in a short bit. Okay, so this is the experiment. So first, let's talk about fish. Right, the effects of polydispersity on small angle scattering. Right, so I'm using an analogy here. Right, we're all biologists, and we have that fish we're interested in. It lives in a complex ecosystem. All right, and so what we do, our first intuition is to take that fish out of its native environment and put it into a fish bowl. Right, we study it in isolation, in situ, in vivo, all right, but it's not a natural environment, but we study macromolecules in the fish tank because they're supposed to be in aqueous environments, all right, and so we have available to us many different methods to study that fish at different levels of resolution, all right. X-ray crystallography remains the gold standard for structure determination at high resolution, right? But again, there's caveats to every experiment. Here, you're taking the fish out of a native environment, putting it in a very artificial one, right? And packing it in, the crystal lattice, right? So we can get very fine atomic details of the structure of that macromolecule, but it's in a very artificial, unusual environment, even though it's aqueous, right? But there's a lot of unusual buffer components and concentration of sample and how it's packed. NMR is much closer to a solution measurement, except you're working with much smaller fish generally in a very crowded fish tank. All right, and there's a lot of good information there, but there's still size constraints with regard to technique. Electron microscopy, the revolution has been amazing for the past several years, especially with regards to resolution and what can be resolved. But again, it's not a solution measurement. Right? You're either vitrifying the sample or you're putting it on a grid and negative stain at lower resolutions. Things that aren't necessarily what's going on in the fish tank. Right? Small scattering is very different from these other three techniques because, right, you have a very low amount of data to parameters. All right, in X-ray crystallography, at 1 angstrom resolution, you may have a data to parameter ratio of like 10 to 1, I think, if I recall, or 8 to 1, right? In electron microscopy with increased resolution, the confidence in atomic position is very high. Same with NMR. Not with this technique. This is a very low resolution technique, so you have to constrain your interpretation of it with the available atomic inventory. Right? Deriving structures de novo, right? Because it's a very low data parameter approach, right? It has to be done with extraordinary caution and a lot of constraints. Right? But when used in combination with other techniques, extremely powerful. By itself, it's a very rigorous test, right, of atomic models, hypothesis A versus hypothesis B, right, and other things we'll talk about, right. So this is a very useful and powerful approach, but we have to temper our expectations about how it can be applied, right. In the best case scenario, to be working with tutorial data during this workshop, right, things like scalps, clams, right, things that are discrete, the MVPs of the world, lysozymes, right, proteins that are very well structured and ordered and compact, okay, no floppy bits, is relatively straightforward with best practices to reconcile the resulting scatter from molecules like this that are homogeneous and monodisperse, right, with their form factor, that is their scattering profile, right. So what do I mean by monodisperse, right? When I say monodisperse, I'm referring to two different qualities that are kind of orthogonal. One is chemical monodispersity, all right? Is it chemically pure? Is it one species chemically? Or physical monodispersity, right? Is it physically one thing, or is it sampling different shapes and configurations in solution? Does it have different flexibility and volumetric properties, right, to make it more polydispersed in a different way? They can happen hand in hand, okay? But for these nice cases, right, these easy cases, right, with best practices, is relatively straightforward to reconcile this resulting scattering with the shape, the form factor, right? Thanks, Rick. Yes?
Monodispersity covers higher order oligomers that are a mixture of monomers.
Yeah, so that's a form of polydispersity. And so the question is, does monodispersity also apply to the description of self-association and aggregation, right? And so those terms go hand in hand in that when you have self-association in a concentration-dependent way, or mixtures of aggregates, I'll show you in the following slides, right? Those are forms of polydispersity that have to be dealt with in the experiment, right? And in essence, that's everything what this slide deck is about. It's that type of polydispersity, right? There are different types of polydispersity. You could have a protein sampling different conformers in solution. And you put that in an X-ray beam and collect the resulting scattering, right? The scattering is a volume-weighted fraction, basically fraction-weighted average of all the contributions of the different conformers in that sample. Right? So you have to account for that in your analysis, right? But if you don't know that up front, you may be misled into what that resulting scattering is telling you. Likewise, intrinsic disorder, right? A large part of each karyonic genomes lack structure, and as a result are extremely flexible in solution. You're going to hear more about that today, right? So say you have jellyfish, right? It's even a more complex issue. That's tractable with best methods, but you have to be aware of it. All right, a priori and experimentally, you should know if your system is intrinsically disordered and flexible in solution, so you can account for that in your analysis of the resulting scattering data. The worst situation is heterogeneity of this nature, where you just have a whole bunch of different types of things going on. This could be because you don't have pure protein or pure nucleic acid with N-1 products, for example. You can have self-association where you have both big fish and little fish, okay? Or you can be working with things that are partially purified. This is even a more complex issue that's very difficult to approach in practice, right? So again, understanding what exactly it is that you're scattering is imperative to your ability to reliably interpret the resulting scattering from the experiment. Okay, for knowledge. So I've been involved in some 25 published small angle scattering articles over the past 15 years, and I think my favorite is this one. All right, because there's a graduate student down the hall at the time named Elizabeth Sweeney who was working on. Was working in Jim Shorter's group at the University of Pennsylvania. They're very much interested in these large hexameric AAA proteins. One's called HSP104. Right, these disaggregators. Right, these are large hexamers in solution, which more recently have been just insights that have been revolutionized by the application of cryo-EM. But at this time, the best game in town were homology models to other cryo-EM structures of medium resolution, and other biochemistry. Right? There was no crystal structure available of HSP104. Via our interactions being on the same floor, Elizabeth became very interested in small angle scattering. She started going on Beamline trips with me. You take her preparations of this hexameric protein that she worked really hard to make, put them in the beam, you scatter it, you go through, do the shape distribution function analysis, the reconstructions, and she gets these shapes. She imposes six-fold symmetry. Said, look at this. There it is. And then I said, hold on, slow down. All right, we want to believe, this is an old reference, I'm old, to the X-Files, the TV show from the 1990s. And this was a poster on Mulder's wall in his office in the FBI. We want to believe that these are real. All right, but how do you know? All right, and I started asking her the hard questions that eventually her committee started asking her. How do you know it's monodispersed? How do you know it's a hexamer, not a pentamer, like some others in this class? All right. How do you know if you're not at a concentration where you have a mixture of oligomers? All right. So she had to double back, and start doing these experiments that we did together. SECMOLs, all right, size exclusion chromatography, inline multi-angle light scattering, sedimentation velocity analysis, right? Convincing ourselves that in these different nucleotide-bound states, we're seeing a single species that can be reliably interpreted from the small angle scattering data. Right? These are all the things you have to do, right? You have to do the work before you get to this point where you can have the cool figure 6. Okay, you have to do figure 1 first. Right? This stuff is figure 1. All right, purity, activity, concentration dependence, aggregation, stoichiometry, right? What buffer conditions is your protein happy in? And will it make the trip to the beamline when you ship it at 4 degrees? Do you freeze it and thaw it? Is it okay? Right? You have to ask all this information. Once I get my data, how am I going to interpret it? What available structural information is there? All right? You have to do these thought experiments well before you even approach the protein in the X-ray beam. Okay, designing your experiment. All right, this is where you have to put a lot of thought in. If you're a crystallographer or if you're an NMR scientist, you're working with domain constructs where you've kind of cleaved off little ends that you think are flopping to prevent lattice formation. All right, those are the first things we reach for when we do a small angle scattering experiment, right? But and then sometimes you have the full-length protein thing in the freezer and you want to analyze that. But you have to ask yourself, do I have an atomic inventory against which I can reconcile the small angle scattering data? All right. What if this is the full-length protein, but I only have structures of this, and this? What do I do in between? How am I going to account for and model those missing bits and pieces? Right? And there's methods to do that and to approach those questions. Do I have the same protein structure? Do I have homology models? Right? How good is a homology model? Right? Is there a predicted disorder? Right? Is it experimentally demonstrated? Right? Does my protein gain structure upon binding to a partner? Right? This is a real thing. It happens a lot in protein-protein interactions and protein-nucleic acid interactions, a gain of order and structure upon binding to a target. So if you have a crystal structure in a DNA-bound state, it may not necessarily match what's going on in the absence of DNA in solution. Right? You have to be open to that possibility. Are there cofactors? Should you have the cofactors present in your scattering experiment? Right? If you have a heme-binding protein and it's only 80% reconstituted. So your scattering can be a mixture of two things. Do I need an excess of cofactor? How am I going to deal with that? Is the protein soluble and stable over time? All right. If you're not making milligram quantities of proteins, it's very difficult to do this experiment because you have to do multiple concentrations and replicates, and you have certain sample requirements at the beamline I will mention. You have to think about these things. Does the protein have post-translational modifications? Glycosylated proteins from eukaryotic systems, all right? They're available in precious quantities. Small scattering data can be very useful in characterizing them, but as part of your analysis, whether it be light scattering, centrifugation, or small scattering, you have to account for that glycosylation, what percentage mass it is, and how that will affect your ability to reconcile atomic structure with the solution scattered. Right? Disulfides, I've watched disulfide proteins aggregate in real time in X-ray synchrotron beams. All right, and so you have to figure out your protein if it's disulfide rich, if it's sensitive to X-rays, if you need to attenuate your beam, or if you need a home source or some alternative approach to applying small angle scattering. All right. How pure is your protein? Did you remove the purification fusion partner? Did you scatter something that has a his tag on it? Are you accounting for that in your atomic inventory? All right. More often than not, we're purifying proteins with affinity tags of some variety, which is cool, but it may affect the native disposition of the biological relevance of the scattering experiment you do within the native protein. So you should account for that in your experiment as well. If you cleave it off, have you fully cleaved it off, or do you have a mixture of cleaved and uncleaved? All right. So how are you doing quality control on the protein that's going into the beam? Okay. And then, of course, the quantity issue. This is very important. Make sure you don't start into a small angle scattering study if you're not making milligram quantities of protein in a robust and reproducible way. So a lot of things to think about. Right. I read a lot of beamline proposals for different destinations across the country, and the biggest issue with beamline proposals in this space is that people aren't hypothesis driven. I want to go and do small angle scattering on my protein so I can understand structure. Right. So anyone who's reading that is going to say, well, you can get high resolution data from other approaches. Why are you doing scattering? Right. But if you write a proposal that's hypothesis driven, my hypothesis is that this protein DNA complex, right, even though it's bent in the crystal structure, is actually extended in solution. And I can actually simulate scattering data and show that. I can experimentally resolve that. Right? If you simulate data, you're more likely to get your beam time because it shows that you've thought through the experiment, right, and how you're going to analyze and process your data. Right? At some resources in the country, like, say, for example, NIST, Center for Neutron Research, or Oak Ridge National Labs, beam time is oversubscribed threefold. So it's extremely competitive to get beam time maybe once a year, right? And so doing things like this is very important, not only for the quality of science that you're doing and the analysis and how you eventually will publish it, but just getting the beam time, being hypothesis driven, applying the things you learn at this workshop in terms of making predictions and showing that the predicted scattering will yield insight into question model A versus model B, for example. All right. Along the lines of making predictions, right? What I love about solution biophysics, right? You can be doing light scattering, analytical centrifugation, small angle scattering of different varieties. All the data, if you do it consistently with regards to temperature and buffer conditions and concentration regimes, should be internally consistent, right? The S value from a sedimentation velocity experiment should be, for the most part, it should reconcile with the result from a small angle X-ray scattering experiment. The diffusion coefficient from a light scattering experiment should reconcile with a sedimentation velocity experiment, AUC, right? That's the beauty of this approach, is that it should be internally consistent and should reconcile with reality. You should be able to take atomic structures and predict all of their biophysical solution properties and reconcile with experimental results. If that's not happening, something's wrong, okay? So this is the benefit of taking atomic structures and back calculating S values, frictional coefficients, all right, and making predictions of what the solution scattering will look like in different, say, ligand or conformational states, okay? And there's so many tools available now, online and for free download, that allows you to do that very quickly and very readily. So I highly suggest that you take advantage of these tools before you even start purifying protein and making sure that when you get the data in hand, right, that you're going to be able to resolve the question that you are asking. Good. All right, let's move on to sample purity. How are we doing time-wise? What time? Okay, good. All right. Purity. All right. I have a core facility at Penn, and I have a lot of collaborators for small molecule scattering and structural biology. And I'm a pain in the butt at this particular step right here. How pure is your protein? If you ask 100 people at some national meeting what 95% pure is, on a gel, they may tell you different things. All right, this is the prototype virus integrates. I'll tell you about a little later. All right. Generally, you want protein that is what we consider crystallography grade, 95 and 98% pure by STS page, or if you're being a real pain, by silver staining. Okay. You really need pure material. Some people do one-step purification. They get 75% pure protein, and they call that pure. Right? That's where I usually say stop, go back, add a step. All right. Typically, you want to be purifying these proteins using 2, 3, maybe 4 purification steps. It could be an affinity step for enrichment, ion exchange, and then gel filtration, for example. Right. More often not, at least 3 steps like that to get to something that's like this. And even this, you kind of stare at it, and you can start drilling around, oh, what's that stuff up there? Will that affect my experiment? Okay. But generally speaking, you want stuff that's pure. Right? And you have to be honest with yourself about this too. So overload the protein in the gel and see if other stuff comes up as minor species so you can estimate exactly how much impurity is in there. All right. If you're working with nucleic acids, more often than not, you're getting them from a vendor synthesized for you. All right. You need things that are either gel purified or HPLC purified, because otherwise you'll have N-1 synthesis species that will contaminate your sample and the annealing with other strands. Okay. You can do this by yourself, or you can have the vendor do it. Obviously, it gets more expensive, but generally speaking, you want to be working with very pure, chemically pure materials. They want these steps in there, right? You want to be very, very laborious and careful how you anneal single strands of DNA together to form a double strand. All right. If you're lazy at the spectrophotometer, right, you're not very careful, your extinction coefficient, you can have an excess of one over the other. To do quality control after the annealing, do a native page gel. Make sure it's one single species and not multiple. Okay. With protein, I just mentioned the crystallography grade stuff. Purer the better. Okay. A lot of times when you're working with nucleic acid binding proteins, right, the silent killer is that sometimes nucleic acid comes along for the ride. So one of the simplest quality control things you can do before or after the sizing step is check the 260-280 ratio. Proteins that say, for example, that pull along RNA, HIV integrase or retroviral integrases are a good example of this. They'll pull along RNA from the bacterial milieu, right? Will have an exaggerated 260 signature where they should have a 280 peak in lone if they're purified properly, right? So be aware of the things you can't say as well. All right, sample quantities and concentration. All right, so here's a different version of that master equation that was shown before, in that it's relating the scattering, the function of Q, right, is proportional to the concentration of the macromolecule, right, per unit volume, the square of the volume of the particle. This term here, as squared, is something called contrast, right? That's the differential scatter between the solute and the solvent, how strongly they interact with the incoming radiation, right? I can talk about contrast a lot here, but it's a thing, all right? The form factor, which is what we're most interested in, right? And then that structure factor, right? How these things are interacting with one another. We're interested in this, all right? And so in our experiment, we want to design it in such a way that we're optimizing for this, right? What concentration of material is scattered, right? In the next page, I include a table from the Jeffries review, which is very useful. What you want to do, it's helpful to look at this equation when you design your experiment, because it reminds you of certain things. First off, the intensity of scattering is proportional to the concentration to mass. So obviously you need a critical amount of concentration of your macromolecule and quantity to get good, robust scattering signal. Right? If you're too low in concentration, it's going to peter out very quickly. You're not going to get a lot of good signal noise. If it's too concentrated, this structure factor part becomes a problem and contaminates your signal if you're trying to get the form factor. All right. So generally speaking, you want to be doing experiments in series of concentrations at the beamline, testing different concentrations and making sure that you understand where you are relative to the concentration dependence behavior of the sample, right? And so in this regard, interparticle interference, that's first determined aggregation, big things, okay, which can really exaggerate themselves in the intensity of scattering because intensity varies as the square of the volume of the particle. Dust particles, aggregates, they have a big impact on the resulting scattering. Okay. More often than not, you're working in this concentration range for a lot of things, but it depends if you're working on a ribosome where you might scatter it around 0.1, 0.2 mg per mil versus, say, an 8 kilodalton tutor domain or lysozyme where you may be up around 20 or 30 mg per mil, right? So it's going to depend a lot on the size of the macromolecule, right? This is a pretty handy rule of thumb for a starting point. You want the mass in kilodaltons times the concentration mg per mil starting somewhere near 100, and then you work your way out, up and down. Right? But that's a pretty good starting point if you're trying to design an experiment. How much volumes? I'm going to go to the next slide here, which I think does a really good job of outlining this. Depending on what the nature of the experiment is. Some experiments require more volume than others. If you're doing an injection on a size exclusion column, it could be 100 microliters. It could be just 50 microliters in a small capillary in a static setup, okay? And then everything else in between. Some mail-in resources might only need 30 microliters, right, sent in a plate. Very important to talk to the Beamline scientists at each of the different destinations. Everywhere is a little bit different. They do, the things a little bit differently with their experimental setup to get to the best results. Good, okay. All right, how do you determine concentration? Right? We are quite, we don't really think hard about this sometimes, right? We'll take a UV spec of it, we'll run a Bradford with a dated sample curve that's saved on the spectrophotometer for someone from 10 years ago, all right? You want to be very thoughtful in how you do your concentration determination because as you'll learn, that information can be invaluable to calculating the molecular mass of your macromolecule, right? There are several different ways you can calculate mass in this technique, and a lot of those measurements, it's important to have an accurate determination of concentration. So if you're working with extinction coefficients from a protein, right? Theoretical ones can be unreliable, upwards of 5 to 10%, right? And that could make a difference if you're looking at the binding of a big thing to a little thing, for example, in the protein-protein complex, right? There is some age-old, tried and true method to experiment to determine extinction coefficients. I highly recommend this article here. If you have a lot of protein, it's not a bad idea to spend half a day at the bench and just figure what it is.
I have a question.
Yes.
Someone asks, how important is it to remove dissolved oxygen buffer? Will degassing help improve the data quality?
The answer is yes, and we'll get to that with the SAXS discussion. Actually, when we get to SEC in the next session, in general, we'll talk about that. But yes, it can have an impact, not only in X-ray scattering, but also light scattering as well. If you're working with nucleic acids, right, the hyperchromic effect, right, DNA with unusual bends to them and so forth, right, DNA digestion and looking at what the absorbance is in the digested form can help create a correction factor that gives you a more accurate determination of nucleic acid concentration, right. Bradford assays and their different derivatives are notorious for being upwards of 5, 10, or even more % off in terms of accurately determined concentration. So do multiple concentrations and replicates and make sure you're working within your assay range, right? Use a fresh assay range. Don't use a same stored one in your spec from someone else that did it 5 years ago, right? But information from I0 from SAXS, from SECMOL's analysis, the RI information, refractive index. There are other different ways that you can check your mass at the time of the experiment. We'll talk a little bit more about that downstream. Okay? But be thoughtful about this. Don't be cavalier and nonchalant because this can haunt you later when you're doing your data analysis. All right. Sample environment, buffer. Buffer is so important in this experiment. The difference between getting no data and getting very fine data is the care you take in getting a good match buffer. Okay. That is buffer identical to what your solute is dissolved into. Right? How do you get to that? There are a couple different ways, right? the gel filtration buffer you use, right, to size your protein in the last step at that stage is a match buffer. Now, say you concentrate your protein down, even so you've washed your membrane, hopefully get rid of the glycerin that's used to preserve it and to store it, and you concentrate your protein. The flow through from the last concentration step is a match buffer. Okay. You can also do quick spin columns. These desalting columns do a pretty good job if you do it very carefully. All right. What you don't want to do is to mix by hand a buffer you think is the same composition and assume it's a match buffer. It's probably not. This technique is exquisitely sensitive to differences in buffer. So if you don't have the right match buffer, this experiment's not going to work. Be very thoughtful about how you arrive at a match buffer for your experiment, even if you're doing SAXS. And I'll mention why that is when we get there. Okay. Make sure you wash these vendor-provided appliances very well before you put your sample in, so you don't have anything come along for the ride, right? As I'll mention later, it's good to use fresh reducing agents with synchrotron measurements, right? Because you want to scatter those radicals, right? Don't make them by hand. And there's a little more forgiving. There's more latitude with SAXS, but still then you still want to be close with your buffer, and I'll explain why that is. All right. Characterization. This is my favorite part. All right. So there are Avogadro's number of ways you can characterize your sample. Some are more useful than others in helping you succeed at doing the small angle scattering experiment. All right. There's a lot of useful information here in these different techniques that you can apply to your analysis of the data downstream. But in terms of getting the best data quality possible and putting the right sample, the right concentration, and the right buffer into the beam, these techniques tend to be much more useful. Explain why. Right now, I'll talk about these three: dynamic light scattering and different versions of analytical ultracentrifugation. And then the next section I speak, I'll tell you more about size exclusion leading up into light scattering, multi-angle light scattering, and then ultimately long X-ray scattering in line with that. Okay. But for right now, let me focus on these three here to close out this part of the slide deck. Okay. Light scattering methods are really, really useful. All right. You don't need a crystal. You don't need high sample concentrations. All right. I can walk up to our DLS with 4 microliters in these little specialized cuvettes, and I can get a diffusion coefficient. Right? It's really handy. Right? Very rapid. It takes just a matter of minutes in some cases, or an hour if you're a patient with SECMOL. Right? And they're broadly applicable to all macromolecules, different sizes and shapes. Material scientists use these techniques all the time for their materials. Okay? And they're, generally speaking, relatively few buffer restrictions. Things like glycerol or really high salt can affect your contrast and the viscosity in solution. But generally speaking, beyond that, right, it's not a lot of buffer conditions. You can really probe the dependence of your protein on, say, the binding of calcium, or the binding of ligand, or the effective pH on the conformation of your protein. Right? But the disadvantage, of course, is that these are all very low resolution approaches. But with regards to quality control, right, and understanding and relating the oligomeric state, the mass, the behavior of your protein to a biochemical activity, all right, or something you're going to do with it downstream, very, very valuable experiments to do. Okay, so what I'm going to focus on here, so the dynamic light scattering in general, is a close cousin of X-ray and neutron scattering, which are really basically just changing. Very nicely shown the theory in the first hour, right? What you're changing is the wavelength the radiation used versus the size of the particle being scattered, right? When you're using dynamic light scattering, you're using wavelengths of light around 658 nanometers, okay, which is generally pretty good to kind of figure out molecular mass, and for very large polymers, angular-dependent information like the radius of gyration. Okay? So right now, let's talk about dynamic light scattering, which is very useful. More often than not, now these days, you will find these instruments sitting at a beamline in the wet lab, right? And you should take advantage of it. So this is a cartoon representation of Brownian motion, right? It's all you basically randomly moving around in the box along with solvent, okay? And what's happening here on very short time scales is that the distance between any two of these scattering centers is constantly changing. Okay, and so what happens with scattering is when you have things that are very close together, the scattering from things that are close together is very coherent, right? So that their resulting scatter is additive, and they increase the signal. That's why the square of the volume of a particle, varies. The intensity varies with the square of the volume of a particle. But then as they move apart, they become incoherent. Signal goes down quite a bit, right? So it stands to reason then that at some time point 0, when the two scattering centers are coherent or close together, they have some intensity. But then as they move apart because of diffusion, right, the rate at which they move around here is a function of their diffusion coefficient, right? The rate of flux from one point to another. The intensity will change on very short time scales, right? That rate of change in intensity is proportional to the size and hence the diffusion coefficient of the particles. So very large scattering volumes have very long time intervals going up and down, whereas small things have very short time intervals. Okay, and we can measure this experimentally. Okay, and so this is an autocorrelation function. We're looking at the correlation of intensity at some very short time point, and then seeing how it decays. That correlation decays the function as it goes longer time scales. Okay? The rate at which it decays, right, is a description of the size of the particle, right, and that can be described by a second-order correlation function. I'm not going to belabor the math here, but by fitting these autocorrelation functions, right, you can get a determination of the diffusion coefficient. Right, so this is basically that function, right, and here is the diffusion coefficient, right. Here's the scattering. Here's H, which would be analogous to Q in small angle X-ray scattering, right. We have some 4 pi sine theta over lambda, okay, and this operator here, all right, this correlation function decay rate. Right? That diffusion coefficient is a very, very valuable parameter of your macromolecule. It can be used a couple different ways, right. The very famous Stokes-Einstein equation, that Einstein, right, helps us relate the diffusion coefficient to a hydrodynamic radius of a macromolecule in solution. Okay? The equivalent of what is basically a hard sphere migrating through some space. Right? So here's lysozyme, and here is that purple line here. I'm sorry, pink, magenta, right, is the hydrodynamic radius of lysozyme, which is essentially basically the largest periphery or sphere you draw around this particle. This is very different, and this will be an important note as RG is introduced to you. It's a very different term from the radius gyration. They're not to be confused. The comparison of these two terms is very useful, okay, because that can be an indicator of shape and spatial extent, right? But they should not be confused because shape affects RG much more dramatically than with RH. Right? And so something important to keep in mind as you move forward and learn about the radius gyration moving forward. Any questions about just dynamic light scattering up to that point? Yes?
Do you think it would be good practice to try, say, a lot of different protein concentrations by ELS in preparation for the experiment?
Possibly. I think on a couple levels. One, just seeing what type of aggregations occurring in kind of native conditions that you can do your experiment at in a dynamic light scatter may be instructive. Maybe you'll see a lot of aggregation. And it will tell you, you know what, I need to do SAXS instead because I need to get rid of these aggregates. I'm not going to be able to separate them from the sample otherwise. It may also tell you that if you test different buffer conditions, there might be one that's much better than the others. Minimizes aggregation over a certain time scale at a certain temperature.
Repeat the question.
I'm sorry. So the question is there utility in doing this experiment at conditions similar to your small angle X-ray scattering experiment? And the answer is yes, there's a lot of utility. And it's a relatively inexpensive experiment with regards to sample quantity. They're doing it in a cuvette format. They have disposable cuvettes now that cost like $3 each that hold downwards of 4 microliters, right? And so in terms of finding the right sample condition, the right buffer, the right pH, salt, concentration range, it could be very useful to kind of probe for aggregation from this approach. However, dynamic light scattering probably has the least resolution of all these approaches we're discussing today. While I can tell you if there's an aggregate there, because aggregates scatter really strongly, it does a very, very poor job at resolving oligomers. Right? You may just see one broad peak, but if you do analytical centrifugation or SEC-mols, you'll see they actually have multiple species in that peak that otherwise might have a modest polydispersity term. So it's very good at detecting big things, but very horrible at resolving small things. Okay. Analytical ultracentrifugation. This is another technique that is very, very useful. All first principles, very old technique, and I'm a big fan of it. Usually my figure one, more often than not, it's a 50-50 between SEC-mols and analytical ultracentrifugation. All right, and so this experiment, the concept is pretty straightforward. You apply centrifugal force, and based on the size and the shape of that macromolecule, it will sediment to the bottom of the cell, the outer periphery. The force is the centrifugal force here. Opposing that force are the buoyant force, that is the mass of the solvent being displaced, and a diffusion force. Right? And so depending on the rotor speeds you choose, these forces can be equal to one another, opposing each other, or the centrifugal force can win out. Depending on how you do the experiment, you can get different types of information. All right. In sedimentation velocity analysis, you're spinning the particle at very high g forces, where the centrifugal force dominates over the diffusion force and the buoyant force opposing it. So it will eventually sediment to the bottom of the cell. The rate at which the particle sediments to the bottom of the cell is a function of its size and shape in solution, and can be described by a term called S, the Svedberg. Right, some units of centimeters to the minus 13th. Right. So here's what the data actually looks like. And so here, this is an older instrument, all right? And inside the instrument, there's a rotor spinning around, and then there's a laser or a monochromometer that's shooting from above and very precisely timed to collect absorbance data across the radius of the cell with a photomultiplier tube below the rotor. Right? So it's actually an incredible piece of engineering. And you can do this for 3 samples at a time in an average rotor that holds 4 positions. Right? And you collect an absorbance. Here's time point 0 of the absorbance as a function of the radius of the cell. Here's the next time point, next time point, next time point. What's happening is, as the solute sediments, the absorbance boundary shifts. Okay. And what you have in the experimental run, several hours, you collect a whole series of these boundaries as the macromolecules. And so,
before you
yeah.
have a question, so I wanted to take now. SAXS be used to check liquid-liquid phase separation of protein in solution?
Oh, that's a great question. It depends on who you ask, right? And so material science, I'm doing one of these studies right now. I'm studying a protein that is a drug target and undergoes drug-induced aggregation, right? And so we've been taking a soft matter approach and characterizing the drug-induced aggregates, and we find actually they're weak gels, right? They drop out of solution. And I've been using neutron scattering, which is just a close cousin of X-ray scattering, to characterize the aggregates being formed, right? It depends on what length scale you're interested in, the nature of the material that you're studying. But in principle, it should be useful if you design the experiment very carefully. But you may need to take a soft matter material science approach rather than a atomistic one. Right, so understanding the length scales and the information you want to get out of the experiment. So provisionally, I would say yes. So we left off here, we were looking at the sedimentation boundaries that are being accumulated. We can describe that data using different extrapolations of the Svedberg equations. All right. Here, we're using something called the Lamb equation to track the movement and the boundaries. Right, the rate of movement, and then using that to model the concentration of different S species in solution to get S values for particles in solution, which can relate to molecular mass, right, and give you a measurement of both size and shape, right. So here's a scattering study I did of a protein RNA complex isolated from a native source. I was only working with microgram amounts of this material, okay, a group 2 intron particle, And here, the particle I wanted to scatter was very monodispersed by multiple methods. It's a nice sharp peak and a very specific frictional coefficient, okay, meaning it was elongated solution and it had a certain S value. Whereas the other stuff that we were considering was actually quite polydispersed, and so we ended up abandoning that particle for its model scattering measurement. Okay, this is an example of what the data would look like. Here are the residuals of the model fit to the data. Okay, and sedimentation analyses are very powerful in understanding solution behavior and mixtures of things that are occurring in that experiment. Sedimentation equilibrium analysis is a different variation of this. You're working at rotor speeds where the centrifugal force is directly opposed by the diffusion and buoyant force in the opposite direction. All right. So what happens is, for a particle of a given size, they'll arrive at equilibrium along a certain radial distance in the cell, right? Resulting in, if you took the log plot of the absorbance of the concentration versus radial position, you get straight lines, right? You can calculate theoretical lines of different oligomeric states of that macromolecule and then compare the log of the absorbance to those lines and extrapolate a oligomeric state. You can take global data, you can take multiple speeds and multiple concentrations and globally fit this data to arrive and test different models of association, including the respective association constants for those associations. This is also very useful because then you understand where KD is in a macromolecular association and relate that to the concentrations you're working at in your scattering experiment. Am I 10-fold above KD? Am I 100-fold above KD? Am I at KD? All right? So that will tell you the degree of mixtures occurring in that experiment. Yes?
Do you consider AUC primer case?
Depends on how strong it is, right? And so I'll tell you that with absorbance optics, you can only work in the micromolar regime of most average proteins. If you're working with nucleic acids, or protein nucleic acid complexes, you can work in the to the few hundred to several hundred nanomolar regime, right? It really is a question of what do your optics allow for versus what the KD is, right? So it can be complicated, right? People want KD, but their KD is nanomolar, but we're using UV absorbance and it's in the micromolar regime. So I'm so far above KD that I can't reliably determine.
Sorry, what would you use for a very technical?
Oh, yeah, there's a bunch of techniques you can use. So if I had this conversation in the course setting all the time, I ask them, well, how strong do you think it is? And then we figure out what you can do. ITC is a great technique, but it's not great, or it's very difficult to do well with very tight associations, unless you're doing a competition format experiment. Microscale thermophoresis, that's a technique where you can get to really tight KDs. It's a relatively new technology, it's popping up, but the barrier to get in and do the experiment is relatively low, right, as long as you have the instrument. And people are using that technique to measure picomolar associations or even tighter, metal binding to a protein, for example. So if you think you have a really tight association constant, that might be one technique to look at. I know SPR has been applied a lot in different systems. I'm not an SPR person myself, but that's a possibility as well. And then, you know what, there's the good old-fashioned EMSA. All right? If you're looking at protein nucleic acid assembly, just an EMSA experiment gets you pretty reliable KD. But a lot of it's understanding where you are concentration-wise in your experiment versus where that KD is. Okay. But with regards to precise determination of molecular mass, AUC is probably the best. Very close second is a multi-angle light scattering experiment. All right, and so this is just a summary of the different types of information and sample requirements you need for these two different types of experiments. Okay, there's the Illuminati in the field of AUC would tell you, you know what, you don't really need equilibrium. You can do a lot of good stuff with just sedimentation velocity, including thermodynamics. All right, and so some very powerful tools and methods coming to be with new types of instruments that really will probably make this an antiquated approach to thermodynamics in solution using AUC. But sedimentation velocity, you can get a lot of information out of that experiment if it's done well. All right, and there's good references out there, a whole other literature and community if you're interested in that technique, which is very complementary to small angle scattering. All right, and then let me close out here. Time-wise, I keep things moving along here. At the beamline, Okay? You've done your best deliberate effort to do all the things right with your sample, pick the best candidate sample to move forward with the scattering experiment, right? But you're not done yet, right? Because there's going to be work to be done. For each of the general user facilities across the country, or a home source, or a neutron source, right? There are beamline specific recommendations about how to do the experiment, right? They'll vary, including sample quantities, exposure times, exposure replicates, data analysis, the accessible Q range, which can be a major determinant in a good analysis. Do you have low enough Q for a big thing? Do you have high enough Q to see the structural features in a conformational change? Signal quality data. They vary from location to location. So it's imperative that you have a good dialogue with the beamline scientists at the respective destination, right? Talk to them about your experiment, what you want to accomplish, what type of quality data you need, what Q ranges you need, right? So they can tell you and give you advice about how to best approach your experiment at their beamline. Right? That conversation is so important because here at APS, right, you have a very good beamline resource in terms of description of the right things to do for different types of experiments that's specific to this beamline. And there's a lot of information there, but just take the time to read it. And it's going to help you a lot in designing your experiment. But take the time to read the Beamline websites, because they've already put these things up and they talk to users routinely in helping them with their experiments. All right. At the Beamline, you have your sample, you're ready to do the experiment. Amongst the first things you should do in preparing your sample to put it in the beam is give it a high-speed spin. Right. Freeze thawing, more often than not, will introduce aggregation to some degree in your sample. Or even just 4 degrees for some period of time, you probably can find aggregation. You might know about that already from dynamic light scattering. Okay? Give it a high-speed spin. Tabletop centrifuge, say 10, 15 minutes at 4 degrees, right? Then use the supernatant for your small angle scattering experiment to minimize the aggregation going into the beam as much as humanly possible. I think now I'd be surprised if I didn't find a nanodrop in an absorbance spectrophotometer sitting at a beamline these days in the synchrotron resources. Just take a few more moments. Take a nanodrop reading, get an absorbance to your sample before you put it into the beam. That concentration may help you later in reconciling your data with the analysis and the determination of molecular mass. Right? Extra little step, but it's worth it. Okay? I told you about this part. Talk to the beamline scientists, not only about designing your experiment, but also analyzing your data as you go. If you're a crystallographer, you have the habit of shooting first and asking questions later. Don't do that here. Analyze your data as you go along. Learn how to use the software at the beamline. A lot of times there are customized scripts that are being used and experimental setups to produce the data from the images to the one-dimensional profiles that you use in your analysis. Check your Guinea A plots, check your data as you go, and make sure you're getting data that you can walk away with and use reliably, right? You're spending a lot of money, time, and effort to go out to these synchrotrons and make these measurements. Make sure you're walking away with good data. Okay? The temptation is to just do one, concentration or one injection and kind of walk away, but it's usually worthwhile to do a couple different concentrations of something, right? Even in the SAXS format where you have kind of a concentration dependent analysis built in, the elution of a particle off of a column, right? But do different concentrations and make sure you're not seeing concentration specific phenomenon occurring, right? And assess radiation damage. The reality is that you're using these very brilliant sources of X-rays that are just macromolecules are not designed to deal with, right? And so try to get a sense of what type of radiation damage may be occurring with your macromolecules using model independent analyses like Kratky plots and other averaging of data that are used now in these beamline reduction software packages. Right? So be aware that this is a real challenge for some systems. Right? Here's a visual illustration from the Jeffries review. Right? If you don't have a radical scavenger like DTT, ascorbic acid, producing a BME, T-set, all right, you can see dose-dependent damage to your protein. Right? Here on a millisecond time scale, this particular beam source. Whereas if you added DTT, some nominal amount, you can attenuate a lot of that damage that's occurring. Okay, glycerol is also very good in terms of radical scavenger. It's something that can attenuate radiation damage. Right, but you don't want to be too high in concentration because that can cause viscosity and pressure issues with the gel filtration column. and also issues with contrast, your signal to noise, right? So maybe it's like a few percent, right? Different storage conditions can have different effects on aggregation and RG. You'll see from doing your Guinea plot analysis as well, as mentioned before. So these are the things to look for in those initial profiles that you get at the beamline to be aware of. All right. This review by Jacques and Trella is wonderful. Definitely read this review. It walks through good data and how different pathologies and data can manifest themselves at different stages of the analysis, even the primary profile, the Guinea plots, shape distribution functions, and other things. And so this is worth a read, understanding why it is that interparticle interference introduces these pathologies into the data that occur, including artificially low Dmaxs, negative P of Rs, and unusual slopes in the Guinea analysis, right? Alongside aggregation, which has the opposite effects and upturns in the Guinea plots, and issues with fitting P of R as well, right? Problems with your sample usually play out with problems in your data. Good. I think that's, oh, and then the beamline, I mentioned additives, right? These are things to be aware of. Detergents, if you're working membrane proteins or things that solubilize the protein, be careful of detergents. They have very complex phase behavior, and they can do their own thing at high concentrations, including forming micelles and bicelles and worm-like things that can really mess up your scattering. Carbohydrate sucrose can affect contrast. High Z elements can affect your scattering results. So be aware of the chemical composition of what it is you're scattering and how the differences in contrast can affect your final resulting scattering profile. The question from before: RNA scatters much more strongly than protein. So if you scatter a protein RNA complex, you have a contrast problem. Your RNA signal is much stronger in that profile than the protein component. So back calculating from a structure was very useful, but if you're calculating ab initio envelopes in that data, there's a major proviso there that you have a skewed reconstruction. Then I mentioned these here, these additives that can be helpful. Don't forget your goods buffers, which are themselves radical scavengers, and phosphate buffers. They're not good radical scavengers, but they're great for biophysics. They tend to be pretty well behaved. Right, I think that's it for this particular segment. Questions I can help with? Yes.
I have a question about concentration.
Yes.
For the scientists in the SAXS workshop, so you know coming in what the but as it passes on to what you call it, the plate count is not going to come off the same concentration.
Sure.
In that case, for molecular weight determination.
Can you repeat that?
So to distill down, so with regards to concentration-dependent behaviors, if you're doing a sizing experiment, do you have to worry about that particular issue, right? And you do on a couple different levels, right? As I'll show you when we talk about multi-angle light scattering, what is in a peak, right? A peak may have a retention time consistent with a dimer, but what gel filtration alone won't tell you is if there's dimers and monomers occurring in that peak, or dimers, monomers, and aggregates. Right? So some of the more advanced methods now available to deconvolute SAXS data actually directly take that problem head on, right? Allow you to get at types of analyses that weren't possible 5 years ago. Right? And so with regards to concentration dependence, you still have a challenge to be aware of going in, because that will affect how you analyze the data on the back end. Does that answer your question, I think?
Kind of. I was more interested in the actual concentration.
Sure.
As far as I
It
understand.
depends on how important molecular weight determination much of a part of aim of the experiment is. So, like Kushol is saying, if you're trying to determine molecular weight to, for example, determine the specialty of a compound, then what he's saying comes into where. But then, yes, concentration independent molecular weight determination will be important if there is no. So ideally, there should be an inline UV or a refractometry system that can determine the concentration just before it goes into the cell. Right now, it's not available in this. There are things that have that. But yeah, there are concentration independent.
I'll give you one example. I'm setting a dimer tetramer in solution, right? Phenylalanine hydroxylase, iamine jacket phosphatase. If I inject it on a sizing column, it may have a retention time of a tetramer, but if I do sec-mols analysis, I can see that it's going to dimer tetramer in the absence of its activator, very clearly across the peak. If you do SAXS analysis, you get a mixture of things, right, in the absence of phenylalanine. But if I do the single value decomposition evolving factor analysis, we're going to tell you about in the second talk, right? You can deconvolute and pull out dimer, tetramer. Do the mass analyses on the dimer and tetramer, get correct masses, and then reconcile those dimers and tetramers with structural models. So there are now, the more advanced methods allow you to deal with these problems head on in a way you really couldn't before. But the problems are tractable.
I have several questions from remote viewers. So the first is, they say they were under the impression that larger size contaminants are more harmful for SAXS than smaller size ones. How much should they worry about contaminants that are much smaller than the size of the target protein?
I'll give you an example. Here I have a protein, a retroviral integrase. We purify using an MXC tag. It's just basically a chitin binding domain. And if the collaborator isn't, careful with the sizing step and the fractions they pick, right, and they include a fraction that has some of that contaminant in there in the gel filtration profile, right? If I take that, concentrate it down, then do SAXS, and do the analysis I just talked about, deconvolution, I can actually pull out the form factor for that small chitin binding domain that otherwise would have been invisible to me or I wouldn't have been aware of. Smaller things, depending on how small they are, still can contribute to scattering depending on what concentration that they're at. Right? If you see them on a gel, maybe be concerned.
So the next remote question is, are there any guidelines in using buffer additives for measuring nucleic acids to protect them from radiation damage?
I think the guidelines are roughly the same with regards to glycerol. Reducing agents probably won't be as helpful, although they do scavenge radicals, but glycerol may be the thing to try first. But also, with nucleic acids and scattering, what you have to be aware of, very particularly, is a very strong interparticle interference because of those phosphates interacting. So having divalence and the right amount of salt in the buffer conditions to prevent interparticle interference from occurring, even with SAXS, is very important because that can skew your data analysis. Doing many concentrations, making sure you have adequate amount of divalent and salt in your buffer conditions. That's it. Any more questions? But all right, thanks, and I'll talk to you more in a bit.